You did a wonderful job. Thank you. Our lab in the College of Pharmacy, which we co-run together with Nora Vasquez, who sits over there, studies the ribosome, the most complicated, sophisticated, and as you can see, the most beautiful molecular machine in the cell. The ribosome is essential for life because it makes all the cellular proteins. And for the past 30-some years, we have been trying to understand how it does it. But what also we're trying to understand is how can you stop ribosome from making proteins. And this is actually very important because crippling the ribosome relates us to antibiotics. There are many antibiotics which have been discovered over the decades which kill microbes that makes us sick by binding to their ribosome and not allowing the ribosome to make proteins. The problem is that microbes are very smart, and very easily they find the tricks to become resistant to antibiotics that we use. And because of that, we humans and them microbes are in the constant arms race. They try to become resistant to the drugs that we have, and we're trying to find new drugs, new better antibiotics with a new mechanism of action. And the best place to look for new antibiotics is nature. For example, insects like honeybees, which, as I heard, are close to the heart of our chancellor, as well as the fruit flies, make their own antibiotics to protect them from the microbes which make insects sick. So the antibiotics that we study from insects are called antimicrobial peptides. What we found is that antimicrobial peptides can get into the very gut of the ribosome. And when they get there, they stop ribosome from making proteins. And in this way, they kill microbes, which we would call the bad bugs. So this is fine, but what is the most amazing is how these antimicrobial peptides kill the ribosome. So to make the protein, the ribosome needs to run on the genetic message. It comes to the departure gate, the start, runs the truck, and then comes to the terminal station, to the finish line. So all the antibiotics that we knew before, they stop the ribosome either at the departure gate or stop it on the trucks while it's run. The antimicrobial peptides from insects do it in a very strange way. They do not trap the ribosome at the beginning, not on the way, but they trap the ribosome when it comes to the terminal station. And since there are many ribosomes which run on the same truck, stopping only one ribosome at the end of the truck takes all the rest of them out of business. It's like if you have a tram line and you stop the very first tram on the line, none of the other trams on the same line can bring passengers to destination station. So we hope that by studying these peptides, we will be able to develop new antibiotics, understand resistance, and learn new things about the ribosome. Thank you.